Ready?
Yeah, hi.
Hello everybody. It's a real pleasure to have here Cian O'Donnell. And Cian O'Donnell has a special background. He has worked with some of the great people, both in machine learning and computational neuroscience. He started doing his undergraduate degree in applied physics in Dublin. And then he did the smart thing that some of us did not do, to visit many labs and works in the States, in Cambridge and in Belgium as a research assistant or a research internship in order to get a feeling for science. And it seems that neuroinformatics won his heart. So he ended up doing a master's with research, which he graduated with distinction from the University of Edinburgh, where he met Matthew Nolan and Mark van Rossum, that were his supervisors. And then he liked Edinburgh so much that after his PhD, he stayed over for a postdoc. But then he moved to La Jolla in the US together with Terence Sosnowski. And after that, he was offered his position in Bristol, where he's for a few years now. We're very looking forward to hear your talk.
Okay, thanks very much, Eleni. I'll try and share my screen now. And okay, is that working?
Yep.
So I can't see the chat or anything now, so I'm going to have to assume that you can all see my screen. Is that good, DJ and Eleni?
Yeah, that works well, yeah.
Looks good.
Okay, great, I'll start. All right, well, thanks very much, Aditya and Eleni, for the invite. Yeah, so like Eleni said, I'm a computational neuroscientist in Bristol, and I'd say our group works on three things. The thing I'm going to talk about today, which is neural circuits in autism, modeling synaptic plasticity and learning and memory. And the third thing is sort of developing statistical methods for neuroscience data. I'll also say that I can't see the chat, so please feel free to ask questions, but maybe we'll in it, but I won't be able to see them to the end, so then I'm going to take questions in, but I'll try and finish early enough so that we have time. Okay, great. So this talk's going to be about neurodevelopmental disorders, and I'll focus on autism, but I think a lot of the lessons are kind of true for other neurodevelopmental disorders like schizophrenia and bipolar disease and ADHD disorders like that. And for a long time, we didn't know too much about the neurobiology underlying these conditions. But that's kind of all changed in the last about 10 years or so with the advent of large-scale genetic association studies. And the kind of key message that's coming out of a lot of them is that, what I've written at the top here, is that these neurodevelopmental disorders, which are, we have these clinical labels based on behavior, turns out underneath they're very genetically heterogeneous. So here's a figure from a recent paper in Cell from a large-scale genome-wide association study where they've taken, I forget the exact numbers, but on the order of 10,000 autistic people and 10,000 typical people, and looked for genes that, or sorry, mutations that are more prevalent in the autistic people than in the typical people. And what's put on the y-axis here is the sort of p-value, so on a log scale. So this is a very significant number, and this is a significant threshold. So let's say that's a false discovery rate of 0.05. So anything up here should be kind of a gene that elevates the risk for autism. So it's worth noting that some of these genes confer a high risk for autism, but they might be relatively rare. And the other way is a gene could confer or a mutation could confer a very low risk for autism, but be quite prevalent, or appear a lot in the population, and that would also give a high or low p-value. So the kind of key takeaway from this paper is that I think even in the title, there's something like 100 or 102 different genes already linked that elevate the risk for autism. Any individual gene only accounts for 1% or less of the total cases. So it looks like this disorder we've called autism is really, at the genetic level at least, totally more like a large set of genetically distinct disorders. And the same, like I said, the same thing's roughly true in schizophrenia. In autism, the variance you can explain with genetics these days is around 80%, and presumably it will climb. With schizophrenia, there's a larger environmental component. It's more like 50% genetic, 50% environmental. Okay, so kind of a natural question then is, if we want to understand and maybe develop treatments for some of the symptoms for disorders like this, what's the best level of investigation to go about it? And as we know, in the neuroscience and in the brain is very sort of multi-scaled, multi-leveled. So a lot of researchers work at the level of genes and molecules, and somehow these things magically wire together to make cells and synapses that come together in neural circuits, and somehow that magically then leads to cognition and behavior. And for a lot of the history of neurodevelopmental disorders, people might have worked out some molecular property at a low level, and then maybe design a drug or other intervention that could hopefully reverse the deficit at the molecular level and hope to treat the symptoms at the behavioral level. There's quite a large gap in between that we don't really understand. So me and other people argue that maybe a more useful level of investigation is somewhere higher up this chain at the level of neural circuits. So why neural circuits in the first place? And I'm going to spend the first half of this talk making the case for studying neural circuits, and I think we're going to argue that it's because of this very prevalent property of redundancy, and specifically 4 of its consequences. So convergence, robustness, dependencies, that's going to be dependencies between circuit parameters, and multiple solutions. And I'll go through each of these 4 in turn to explain what I mean by that. Before that, I should say that all of this is almost directly lifted from the ideas that Eve Marder has been pushing for many years. So if you've probably heard of Eve Marder, but if not, her group have thought about these issues very carefully using a simple preparation from the stomach of a lobster or a crab, where they know the purpose of the neural circuit. It's to generate some oscillations that help the stomach chew food or sort of dissolve the food. And she and her trainees have thought very hard about this outside the context of thinking about brain disorders. So what we're trying to do is import some of her and other people's work into thinking about brain disorders along these four aspects. So I'd recommend this nice review paper she wrote, and many others. So how should we, so just to back up a little bit, I'll come back to those 4 aspects of redundancy. But first we want to think about why neural circuits in the first place. And we're going to suggest that a good way to think about neural circuits and brain disorders is to think of them as performing computations that we can think of as an input-output function. So here by function, I kind of mean more in the mathematical sense of a mapping from an input to an output, rather than in the more common language in biology. A function of a circuit would be something like, what does it do? What's its purpose? So let's say V1 does processing for vision, loosely speaking. Here we're going to be a bit more in line with mathematical usage, which is you've got a circuit that takes some input and transforms it to an output. So in the case of a neural circuit, let's say your favorite bit of the brain, the thing that it sees as input are spike patterns from other bits of the brain. So you can think of this little egg shape here as the space of all possible spike patterns. So that's a spike pattern, some firing of some set of neurons. That's another possible spike pattern, and so on. And then the circuit it sees these as inputs, and then it generates some other outputs that live in a different space, depending on how many spikes it can have. So, and what it does is map, for every given input spike train, it'll generate an output spike train. And so roughly speaking, there's a one-to-one mapping. Maybe if you include noise, you could think of this as a probability distribution. Okay, and that's what I'm going to say the sort of computation is, turning inputs into outputs. And to put some cartoon mathematics on it, we can think of if we call the inputs, or describe the inputs with some random variable x, and the outputs of the circuit as another variable y, then we can think of the circuit as implementing a function f of x, mapping x to y. And for neural circuits, we know that, or what determines this function, we can think of as being set by some parameters of the circuit. So here I've called theta, kind of in line with the statistics and machine learning terminology. theta here is you can think of it as a big set of parameters. So that would be for some part of the cortex, LER2-3 somatosensory cortex, that would be your list of all your cell types, your PV interneurons, excitatory cells, the synaptic connectivity, the ion channels expressed in the neurons, the properties of cells like the input resistances, anything you can think of be a parameter. That's something that could affect this input-output mapping, something that can affect the function. But it's not the function itself, it's the sort of measurable components of the brain. And finally, just to layer it on top of that function, from the point of view of the brain as being commanding the body, and also from an evolutionary point of view, some of these functions are going to be more useful than others. So what we're going to say is that each setting of a function is associated with some utility that is itself a function of the function, but also depends on the statistics of the input to that brain region. So, for example, if your primary visual cortex, you might be implementing some function on your inputs that are coming from the thalamus and processing sensory signals about the world. But then if somebody closed your right eye or injured it, then the statistics, the P of X of your inputs would change. And now maybe the old function you were doing wouldn't be as useful anymore. You might have to adapt the function to bring back up the utility or the usefulness of the function. So for that reason, I'm going to be super general and just say that the usefulness of the utility of the function depends both on the function itself and on the inputs that the circuit's seeing. So that's all quite abstract and vague, but just bear with me. So kind of in the existing neurodevelopmental neuroscience literature, a lot of the people that work on, especially that work on animal models of these brain disorders, typically take usually a mouse. Find a mouse with a genetic mutation that mimics one of the mutations observed in the human population, and they'll go and measure properties of the brain. Typically that's these, what I'm going to call components, so things like how many PV interneurons are there, or what's the strength of the synaptic connections. And they're going to ask that, they usually ask this question: how are the thetas changing? What's the difference between the parameters in a wild-type mouse versus a genetic mouse model of autism or whatever? The functional viewpoint's a bit different. It's saying what's the sensitivity of the function with respect to each of the parameters? How does the function depend on each of the parameters locally? Or maybe a different question is, let's compare the function implemented with parameter set 1 versus the function implemented with parameter set 2. So maybe parameter set 1 is the configuration of your LER2-3 somatosensory cortex in a wild-type animal, and this is your configuration in a mouse model of autism. But the key thing is you're comparing the functions directly rather than the components. And the reason we think this is useful is because of, or this is a better way to go than looking at the components directly, is because of this property of redundancy. I'll get into in a minute. So the 4 consequences that I listed already of redundancy. Well, I'll go back up a little bit. The generic idea of redundancy is that there might be many or multiple configurations of thetas that give you effectively the same function at the circuit level. So it's sort of a many-to-one relationship. That's all that I mean by redundancy. And again, like I said, this idea has been fleshed out by Eden Marder and colleagues in quite a lot of detail. So within that generic idea expression, doesn't really matter for this purpose. And then in that space, we're going to lay a contour map down for the utility. So there's some level where there's, the circuit performs at a, I'm not going to use the word optimal, but a high level of utility, somewhere in the middle. If we have too much or too little of either of these 2 component settings, then our utility drops off. That's the generic idea behind convergence. So let's say all the typical people, if you measured their sodium and potassium channel expressions, each of these dots might land somewhere in this high utility region. But equally, if we measured the same things in mouse models of autism spectrum disorder, they might live in a slightly different position in this 2D parameter space, but it might still have a high utility. So from that point of view, these changes might be real. The genetic mutation might, via development, be leading to changes in the properties of this brain circuit that you can measure, but it might have no effect or a small effect only on the utility of the function. So it's sort of benign from the circuit function point of view. That's entirely possible. And that's the kind of the key idea underlying when I'm going to use the word convergence or redundancy. I should say we're trying to write up a review paper on this at the moment. So if you have any better words or objections to the words that we're using, please email me or contact us after, because we kind of want to get it right and we don't want to confuse people. And the other thing to point out is that this is a 2D cartoon. In reality, like I was trying to imply, the real brain circuits probably have thousands or millions of parameters. So this picture, just this simple bump that I'm drawing here, is probably massively under representing the complexity of the actual landscape. And hopefully this idea is not controversial at all to anybody that thinks about machine learning or deep learning. This would be like your cost function or something like that, as a function of some parameters of your network. Okay, so the next property is going to call robustness, and by that I mean I've drawn a new utility function that's a bit squashed. So now this means that the utility is more sensitive to some parameters over others. So if we start here at the peak of utility, if we move a small change in the vertical direction, changing θ2, we get a big change in utility. So the circuit utility is very sensitive to changes in θ2. However, for θ1, it can kind of handle big changes along this axis. And that means it's robust to changes in θ1. So if typical people, their parameters sit somewhere spread out in this region along the axis of robustness, so it can handle a bit of variability along this axis without changing the utility too much. Maybe the autistic mouse models or people's brains live somewhere else in this space, and they might have a change both in θ1 and in θ2. But if you were to measure, the danger here, consequence of this is that if you were to measure both θ1 and θ2, you might see a big change in θ1 and a small change in θ2, and a big change in the overall utility if you could assess it somehow. Maybe the mice aren't as good at some visual discrimination task or something. You might attribute that change to the big component change. You might attribute the circuit utility deficit to the change in θ1, whereas actually it's mostly been driven by a change in θ2. Second problem arising from this is if you were only to measure some of these parameters, so for example, if you measure θ1 but not θ2, you would see this shift from these blue dots along the x-axis to about here. You would measure that difference. And that might be a real difference, but it's not, if it's not contributing to the circuit function utility change, then it's effectively benign. So you might, the tricky thing is that it might actually correlate with the circuit function. So for example, if you compare it across animals, which ones have lower or higher variability, then you might observe that change, as I've kind of implied on this axis here. So then if you were to design a treatment to reverse that effect, some sodium channel blocker to bring the green dots back over here, whoops, slide the green dots back, slide the green dots back over here, you would be correcting the change in θ1, but you'd still have a low utility. And I think that's the bigger danger, because we're never going to be able to measure everything. There's thousands of things changing, and we don't know the joint dependencies. It's going to be very hard to imagine how to measure everything simultaneously. So there's always going to be hidden parameters that might be contributing, actually driving the circuit function changes that we can't identify, or it's very hard to identify. Third thing on the list was dependencies. So now I'm just kind of going to rotate the utility function so it's got some dependence between θ1 and θ2. So now if θ1 and θ2 are both high, that's okay from a utility point of view. If θ1 and θ2 are both low, that's okay from a utility point of view. What it's bad is if you're up here or you're down here. So there's high θ1 and low θ2, or vice versa. So there's some dependency in the sensitivity between parameters. And again, we think from Eve Marder's work and others that this is likely to be quite common. So maybe now the typical people and the ASD people live somewhere on this axis. So they end up having similar utilities, but big changes in both parameters. Very similar picture to the last slide, that measuring one of these properties on its own would be misleading, because if I tried to correct now either of the parameters individually, so if I tried to reverse the deficit in θ1 or θ2 by sliding the green dots down here or sliding the green dots over here, I'd actually be making the circuit utility worse rather than better. And the challenge is understanding the shape of this landscape is always going to be hard, and it only gets harder and harder the higher the dimensionality of the parameter space. And finally, the most extreme form of redundancy is the idea of multiple solutions. And what's that? That just means that there may be different sort of islands in this parameter space that have high utility. So maybe all the typical dots live on the peak of this island over here. There might be a valley of utility in the middle, and then all the ASD people might live somewhere on the other side of the valley. Again, in statistics, this shaped utility function or cost function is called convex, and that just means that the points in between two solutions are themselves, like if you interpolate between two solutions, you don't have a continuous line of good solutions. And that's very tricky because there's so-called local optima in this landscape. And the knock-on consequence here is that, or a knock-on consequence of this is, maybe for these green dots, maybe the easiest intervention to correct the deficit, bring the circuit utility back up, wouldn't be to try and reverse the original deficit, to slide the green dots back over to the blue ones, but maybe there's another intervention that could slide down here that would improve things for the ASD mice or people, but the intervention wouldn't necessarily just be a direct reversal of the original change. So that's kind of another lesson, and that's a slightly more positive note, because it means that there might be multiple routes out of this bad config. Well, I use the word bad configuration. I want to be careful and don't want to imply that the ASD dots are always low utility and the typical people are always high utility. That's not the case. We know that ASD autistic people are just different. However, there are some symptoms that can be debilitating in some fraction of the population. Bad sleep, low IQ in some cases, and sensory hypersensitivity to make life uncomfortable. So maybe we can think about treatments for those symptoms, but we don't necessarily want to cure autism or something like that. That's not general. That concept doesn't generalize to other neurodevelopmental disorders. Okay, so just to summarize, what we're trying to argue for is that from the functional point of view, the utility of circuit function is all that ultimately matters for cognitive symptoms. It doesn't really matter what configuration the brain circuit is in, as long as it's doing the right job. And given that, circuit component changes are important only via their effects on circuit utility. However, that relationship is complicated by this property of redundancy. We've gone through 4 aspects of redundancy, and they all have slightly different lessons and predictions for what we might see in animal models, and presumably also in humans, which is harder to measure. And then finally, I'm going to make a plug for computational modeling as a useful way to address this problem, because not just that I would say that as a computational person, but like I said, if there's relationships between the parameter effects on circuit function, it's going to be very hard to dissect out all the contributions in an animal model or just by purely observing. So even if you can do your intervention, it's very hard to know for sure that the effects you see are directly contributing to the circuit function deficit. With computational modeling, you're God, you're in control, so you can directly change each circuit parameter in your model and ask how does that change the circuit function. So of course, models aren't real, they're wrong, but they are a way to put your money where your mouth is and say, if I think the sodium channel underexpression is the contributing factor to poor visual discrimination, then I should be able to build a computational model where I can replicate that. And that will help me sidestep or argue against the possibility that that real parameter change is just sort of benign from the circuit level. So it's kind of a good way to build a logical relationship between component function and utility links. Good. So that's the first half of the talk. And like I said, we're hoping to write up a review paper, or we are writing up a review paper about this, and welcome any comments. So we'll move on to the second half, which is implementing or testing a circuit function theory for autism. And so it's kind of loosely related to the first half, but it's a purely empirical study. So autism is a brain disorder traditionally diagnosed by deficits in social communication and poor maintaining social relationships and repetitive behaviors. These days, they've also added to the diagnostic criteria, in the US at least, sensory processing deficits, so particularly sensory hypersensitivity or hyposensitivity. So sometimes autistic people don't like close touching or loud noises or bright lights. And one possibility is that those sensory processing abnormalities compared to typical people are upstream of the more sort of abstract cognitive changes. So for example, if you have difficulty processing sounds as a child, maybe it makes it harder to understand language and therefore harder to communicate with people, and maybe a disengagement from social situations. That's a theory for how sensory processing, their learning development could affect these more abstract symptoms. So one nice theory that I like, because it's a circuit function theory, is this idea of neurovariability. So here's a figure from a study from Elizabeth Milne in Sheffield, or was in Sheffield anyway, published nearly 10 years ago now, where she measured with EEG, scalp EEG, she measured the event-related potentials in response to a flashing or a visual stimulus. So here, x-axis here is time, and each row is a different trial, and the color encodes the amplitude of the averaged event-related potential in the scalp. And when she measures, you see this sort of very standard event-related potential, and she can measure both the peak amplitude and the trial-to-trial variability is quantified by the standard deviation. When she measures that in ASD people, she gets some high level of variability, and the trial-to-trial variability is lower in typically developing people. So in this sense, the trial-to-trial variability is higher in autistic people's brains in response to sensory sensitivity. And the rough idea is that there's more variability that might make it harder for downstream brain regions to process that signal, kind of adding noise to the signal. At least that's my interpretation. Here's a follow-up study showing the same thing, basically, but using fMRI and 3 different modalities. So they did visual stimuli, auditory stimuli, and somatosensory stimuli. This is the following year by Redundancy. And in orange, they have the mean responses for autistic people compared to typically developing people in control. So the mean responses are, the amplitude of the mean responses in response to these visual, somatosensory, and auditory stimuli are the same, whereas the trial-to-trial variability, standard deviation of trial-to-trial responses, is higher in each case, by 20% higher in autistic people. So I like that idea, and we wanted to follow up and basically ask, can we find the same thing in the cellular level in animal models of autism? And I met this guy at Landinstein, who's now got his own group, and he said he was going to neuroscience conferences for years and asking experimental people to work on animal models if they'd ever looked at the variability, and they kind of kept fobbing him off for years. So he seemed happy that somebody was finally trying it. So the questions we're going to ask following this theory are, can we find the same thing at the cellular level in animals? So is there excess variability in neural responses to identical sensory stimuli? If I show the same stimulus multiple times, do I get more variability in the autistic mouse model? For a related but different question we're going to ask is, how stable is that brain response, neural population response to sensory sensitivity across days? So if I present a sensory stimulus and record it in an animal brain, wait a few days, show the exact same stimulus, and how similar is the brain response to that stimulus? And is that similarity higher or lower in autistic mice models? And then finally, another version of the variability idea is that maybe there's heterogeneity in brain properties across or within individuals. So we're going to look at whether there's heterogeneity in cellular firing rates within individuals is different between wild type and mouse models of autism. This one caveat with the variability literature, which is almost all exclusively in humans, is that originally it was kind of defined in this number 1 sense, but some papers think of it in number 3 sense. The second sense is potentially under the same umbrella, but it's different. So I just want to be clear about the differences. So all of this work was funded by a grant by the Simons Foundation. And all the experiments I'm going to talk about were done in the lab of Carlos Portero-Cayo in UCLA, done by his 2 postdocs, Nazim Kordugli and Anand Suresh. And then all of the data analysis was done by my postdoc, Beatrice Mizusaki, here in Bristol. Okay, so to explain exactly what we did, we didn't exactly look at mouse models of autism. We looked at mouse models of a different brain disorder called Fragile X syndrome. And the reason for that is, well, I'll explain first what it is. If you haven't heard of Fragile X syndrome, it's a very popular brain disorder for neuroscientists because we've known the genetic origins of it for many decades, I think since the 50s. And there's been a mouse model of the disorder since 1994. So there's quite a lot of literature built up on the properties of the changes in the mouse brain as a result of the genetic perturbation. Whereas all those other genes I mentioned at the start were only really discovered in the last 5-10 years. So there's way less known about their effect on brain function and development. So for that reason, it's kind of especially appealing from a computational point of view. There's lots of nice data to constrain our computational models and thinking. So what is it? It's genetic neurodevelopmental disorder due to silencing of the FMR1 gene, which is on the X chromosome, which is why it's called Fragile X syndrome. Because it's on the X chromosome, it's twice as prevalent in males compared to females. It's sort of relatively rare, but still prevalent. The key symptom is intellectual disability. Most of the people with this disorder have a low IQ. There are other more variable symptoms, such as sleep problems, elevated risk of autism. So this is the bridge to autism. Sort of, if somebody has been diagnosed with Fragile X syndrome, about 1/3 of those people also are diagnosed for autism. So there's overlapping social communication deficits. There's also a higher risk of epilepsy. About 10% of people have epilepsy, which is much higher than the 1% prevalence in the general population. There's other physical characteristics, elongated forehead, so-called pronounced ears. These are quite minor and thought to be benign. Surprisingly, this is the largest single genetic known cause of autism. So of all the people with autism, about 1% of them have Fragile X syndrome. And some of that might just be due to diagnosis, but it's a small fraction of all the people with autism, but it's the biggest single known cause of autism at the moment. But the other genes account for less than 1% each. So we're going to study it in this mouse model of Fragile X syndrome and FMR1 knockout. And we're going to look at the particular bit of the brain in somatosensory cortex that's responsible for responding to whisker stimuli. So the whisker pathway, there's a hair follicle here at the base of the whisker that has a nerve that projects all the way to thalamus, then projects to the brainstem, then to the thalamus, and then directly to layer 4 in the somatosensory cortex. And we're going to look at the next level of processing, more superficial in the cortex, layer 2-3. So roughly speaking, the purpose of these cells is to process sensory stimuli evoked by the whiskers. And so here's I'm going to do the data that was collected by Nazim and Anand in Carlos's lab, but it's done with 2-photon calcium imaging. Here's one of the extremely raw videos that I naively turned into a PowerPoint background. And you can see each of these blobs here is a different neuron. So that's a cell, that's a cell. This is a top-down view of somatosensory cortex. These cells have been infected with the calcium-sensitive protein. So when the neuron's active, the cell lights up. The activity is very low-pass filter, though, because the dye is on the order of 2 seconds, sort of impulse response function, if you like. So here's our strategy. How do we use this to address the questions I talked about in the last slide? They're going to take 2-week-old animals, P14 meaning 14 days old, record some spontaneous activity when the mouse is awake but not doing anything. We're going to twitch the whiskers, grab the whiskers in a bundle and wiggle them at 5 Hertz for 1 second. Wait 3 seconds, do it again, repeat that 20 times. So we have sort of many trials, 20 trials of 5 Hertz stimulation. Then take a break, do it again, but wiggle the whiskers at a slightly different frequency, 10 Hertz. These two frequencies are within the natural whisking range, which is about 5 to 10 Hertz or 8 Hertz or so. So it's discriminating these two stimulation frequencies is fairly natural task, we think for the animals. And then finally measure some more spontaneous activity. So we're going to record the activity of the neurons in response to each of these conditions. Then in Carlos's lab, they put the animal back into the home cage, wait 5 days till it's 19 days old, take it back out, repeat the exact same experiment again. So we can ask the question about whether the cells are responding differently 5 days later. And then finally, do the whole thing again, but in FMR1 knockout animals, and we do about 8 to 10 animals in each group. So these are quite young. If you're not familiar with mouse ages, 2 weeks to 19 days is still quite early in development. So we're hoping to see there's a lot of changes are happening in the brain naturally anyway during development at this point. An adult mouse is something more like 6 weeks older, or probably older than that, maybe 2 months, 2 to 3 months old. They don't even open their eyes until P11 or 12. So these are really little pups that aren't doing too much. So a little bit more details for any neuroscientists out there. So we have these two genotypes at the 2 different age time points. The dye they're going to use is GCAM6S. So it's S for slow, meaning it's got about a 2-second response after a single spike or a small number of spikes. So they inject that with a virus at 1 day old. And then the reason for that is it takes about 2 weeks to express, so they have to come back at P14. Implant a cranial window on the top of the mouse brain, meaning open up the skull, implant a window at P12, give it a couple of days to recover, and then do the imaging at P14. Typically, the big challenge was finding cells at both P14 and P19. So the guys had to re-identify the same neurons at the 2 time points. That turned out to be incredibly tricky. It's becoming fairly routine to do this in adult animals, and you use things like the blood vessels as landmarks. But this early in development, the brain's changing a lot, so the blood vessels move around, the brain grows in size, and turned out to be a massive effort from Nazim and Anand to do this task. And as far as I know, nobody has ever done this in animals this young of any genotype, mice this young, I should say. So they've eventually matched between 13 and 95 neurons in each animal between the 2 sessions. And the imaging frame rate is 8 Hertz. So you're not going to see very high frequency spike timing events here with this recording method. You're sacrificing that for imaging all the neurons in an unbiased way in your field of view. Okay, so they record these movies like the one I showed you in the background a minute ago. Then with some automated software, they can segment out individual neurons, draw little circles around each cell, take the average fluorescence in each of the circled pixels, and plot a fluorescence time series for each neuron. So each row here is a different neuron. That's neuron number 1. See these little blips correspond to when the neuron responded. And that's the data that the Los Angeles group sent to Beatrice. And then Beatrice, in some cases, analyzed this directly, or in other cases, passed this data through a spike inference algorithm to try and sort of remove the effect of the dye and to infer when the individual spikes are happening. That part is notoriously tricky. We know we can't get every single spike. We're probably getting half-ish of spikes. The activity is very sparse in this bit of the brain. Usually, if you twist a whisker, only a neuron might respond only with 1 or a few spikes. So it's fairly optimized case for calcium imaging. But we know it's still partial information about the true activity of the neurons. So we'll see how we do. So what happens? We get data that looks like this. This is an example of the trial-to-trial variability we get. So this is a case with 19 neurons, so each neuron's on a different row. And I've color-coded the number of spikes that the neuron emitted on each trial. So each column corresponds to a different trial. So in this case, there was 40 trials at the 5 Hz whisker twitching. And you can see that there's a lot of trial-to-trial variability. Some neurons are more active than others. This is a very active neuron. These two neurons were barely active at all. If we compare that to the same neurons recorded in response to a 10 Hz whisker wiggle, it looks similar but not the same. So for example, this neuron that liked the 5 Hz stimulus a lot doesn't respond as much for 10 Hz. These neurons are, on the other hand, are more active for 10 Hz whisker stimulus. So there is some information in here about which stimulus it was. There are only two conditions, and you can see that the brain is somehow representing the stimulus in the spike patterns coming out of these neurons. So I'm going to quickly now show you the results. So the first thing we measure, it's not one of the questions we want to ask, but it's kind of the obvious thing to measure, is what's the mean firing rate? So we've got 4 conditions, 2 spontaneous activities, the 5 Hz stimulus and the 10 Hz between the 2 ages, P14 and P19, in wild-type animals. And the average activity rate, so roughly speaking the spike rate, here each pair of dots is an individual animal. Didn't really change much between spontaneous activity and evoked activity. So that's different to, for example, monkey visual cortex, where the firing rates go up a lot on stimulation. Not really much change here across development or across stimulus conditions in terms of average firing activity. The knockout animals, it's a similar story, except there was slightly higher firing rates in P14. So there was significant elevation of firing rates in the P14 knockout animals compared to wild type at the same age. And that's a bit consistent with previous literature, although there's a big debate in the field about maybe some of the sensory hypersensitivity, the fact that the animals respond more to sensory stimuli might be due to these elevated firing rates. So we see some evidence for that, but it's quite small, I would say. Another thing to measure is the synchrony or correlation between neurons. So if we measure for every pair of neurons, measure how coactive they are with the Pearson correlation coefficient, compute that for all pairs of neurons in an animal, we get 1 dot. And if we measure that across these 2 ages, P14 and P19, and in wild type and knockout, we see that there's not much difference between genotypes, but there is a decrease in correlations across development. And that's, again, totally consistent with previous works from Carlos's lab and others in the field. Early in development, there's these big waves of synchronous activity where all the neurons are on together and off together. And that translates into higher correlation, pairwise correlations, so it decreases during development. But we don't see that. We had previously published, and Carlos had previously published, higher correlations in knockout animals in young case, and we might see that again here, but it's quite a minor effect. So now there are 3 questions. So first, we want to ask, what happened with trial-to-trial variability? And the way we quantified that was for each individual neuron, we measured got these 40 different trials for each whisker stimulation. We measure the spike count in that 1 second window after the whisker. Neuron spike 1 times, or 4 times, or 0 times, or 7 times. Measure the standard deviation of that spike count across the 40 trials. That gives us a number, measure variability for that single neuron. And take that number, average it across all the neurons in a single animal. That gives us a single dot. And repeat that for all the animals in the group. And what we can see is that the standard deviation, trial-to-trial variability in spike count is higher in the knockout animals than in the wild-type animals at the younger age. Definitely the younger age and maybe at the next age. So this p-value I've written up here is kind of the ANOVA ignoring age. This is, I think, the first example of replicating the human level results at the cellular level in an animal model of one of these in autism. And it's not necessarily the case that should follow from the EEG or fMRI data, because that's a macroscopic measure where it's averaging across many millions of neurons. This is at the single neuron level. It didn't necessarily have to follow. So we were quite happy to see this result. Next couple of questions were how stable is the representation across this 4 or 5 day period? And we'll measure that in a couple of ways, or 2 or 3 different ways that I'll explain now. First is how stable are the individual cell firing rates? So if a cell fired at 4 Hertz in response to the stimulus on the first day, does it also respond to 4 Hertz on the next day? Yes or no? Or is it higher or lower firing rate? If we take all of the individual neuron firing rates, we get like an array or vector of them, compare the similarity of that vector across the 2 days, we get similarity measured with the correlation coefficient across all the neurons. Do that within an animal, we get a measure of the similarity. So 1 would be perfect match, 0 would be no correlation. It's quite a lot of heterogeneity across animals, but no obvious difference between genotypes. So there's not like the firing rates are more stable in the knockout animals versus the wild type animals across this 5 day period. However, if we do the same thing with pairwise correlations, so if 2 neurons are coactive on the first day, are they also coactive on the second day? The similarity across the time points is quite low, so it's well below 1, but it's statistically above 0. And it's also more stable in the knockout animals than in the wild type animals. So there's more stability in the correlations in the knockout animals than the wild type animals. And that's the opposite of what we hypothesized. Don't really know why. A different way to measure the stability of the representations, more trendy way, is to fit a decoder to the activity. And by that I mean you're going to ask the question, if use a statistical model to learn the relationship between the stimuli, so only 2 stimuli, 5 Hertz or 10 Hertz, and learn the relationship between that and the neural responses. And then you give the decoder a new set of neural responses that it hasn't seen before and ask it to predict, did that come from the 5 Hertz stimulus or the 10 Hertz stimulus? And it's a binary task, so chance guessing will get you to 50%. When we do that, when Beatrice does that, she trains a decoder on the data from day 14 and tests it on the data from the same day. She can guess correctly about 70% of the time, both in wild type and knockout. And it doesn't look like it's any different or substantially different across genotypes. However, if to look at the stability, she can take the same decoder she trained on day 14 and ask it to predict the stimulus from activity from the other 10 point from the P19. And it drops surprisingly below chance. We still need to figure out exactly why, but there's not obviously a bigger drop in the wild type versus the knockout. So from this measure, it doesn't look like the knockout or wild type stability is any different. She can play the game the other way, where she can train the decoder on data from day 19 and ask it to interpret data from day 14. In this case, the decoder in the knockout animals did better than the on the wild type animals at this P19 time point. That might seem to go a little bit contradict what we found before, where the trial-to-trial variability was higher in the knockout animals. You might think if trial-to-trial variability is higher, then the decoding quality should go down. And this is going against that idea. Should caveat that result was previously more obvious in the P14, whereas this is in the P19 animals. However, the ability of the decoder to separate these two stimuli depends not just on the trial-to-trial variability, but also on how sort of far apart the representations are. So you could have more variability in the knockout, but as long as it's separated the 5 Hertz and 10 Hertz stimuli better, then the decoding quality could also go up. So that's not something we would have predicted. And then, something I forgot to mention is this particular decoder assumed the neurons were independent. So if the decoder knew about the correlations between neurons, in principle it could do even better because we know that there are correlations. And particularly, we saw that the correlations were more stable in the knockout than in the wild type animals. So a decoder with correlations might show a bigger stability in the knockout animals than in the wild type animals, but we haven't completed that yet, that analysis. And finally, The third thing we looked at is how heterogeneous are the neurons within an individual animal. And to measure that, we took a simplest measure we could think of, which was get the mean spike firing rate of each neuron in an animal. So let's say 20 neurons in one animal. Get the standard deviation of that, measure how spread the firing rates are inside a single animal. That gives us one of these dots. And compare that for the different stimulus conditions and different ages. And what we can see, at least by eye, is that the heterogeneity in neural firing rates within an animal is higher in the knockout than in the wild type. What does that mean? We don't know, but presumably it's got some knock-on consequence for the neural code, so how the brain is representing these stimuli. So we're still digging into what exactly is driving this increased heterogeneity, and presumably it's got some contribution or effect on the decoding results we had in the last slide. Good, so to summarize this part of the talk, what do we find? We find increased trial-to-trial variability of single-cell spike counts, at least in the younger age point. We're adding more mice to the later age point, so we might also find the data were at least trending in that way for P19. Higher decoding accuracy in Fragile X mice, which we didn't expect, especially given this result. Correlations are more stable across that 5-day interval in the Fragile X mouse models, and increased within animal heterogeneity in Fragile X. So all of this is kind of pointing towards some perturbation to the type of neural code and the stability of the neural code in these Fragile X mouse models. But we didn't in any way investigate what cellular changes were driving that. So in this case, the input to our circuit is the stimulus, and the things we're measuring is the neural activity. We don't know where that change, those variabilities arising from. It could have come anywhere in the circuit, all the way from the whisker follicle to R23. We don't know that, but that's kind of our view for why look at the circuit viewpoint rather than the cellular component viewpoint. And just to finally point out that we looked at this one tiny slice of the brain. Is that going to be the answer to understanding autism? Of course not. But because these are largely genetic disorders, we hope that some of the circuit level changes we see in this part of the brain are replicated across different bits. So if we can find some high-level principles, maybe they will generalize. Or at least there's some hope that they will generalize and lead to thinking about treatments, if that's what we care about. Okay, thanks. I will stop sharing my screen if I can figure out how to do that. Oops.
Yeah, thanks a lot, Cian, for the wonderful talk. We have a few questions waiting for you, and they've voted up. So I'll start with the, maybe I'll just invite Nada on the screen for the top question. Let's see if they can come on. If not, can you also see the question? Yeah, okay. Yeah, yes, there you are. Yeah, go ahead.
So yeah, my first question was how the utility functions or the landscapes are computed in that case, because you mentioned like some landscapes or presumed some landscapes for the utility functions. So how these landscapes are determined or computed, basically?
Excellent question, and I don't know. It's quite possible that it's not doable. I mean, so I try to argue that these things kind of implicitly exist in the brain. Some circuits configurations are better than others. How you would ever measure that, I don't know. All we can measure is performance of some task, really. So it's up to us as neuroscientists, to define the task. Whereas in something like supervised deep learning, the task is explicitly defined, and you can go in and evaluate this cost function explicitly because you can write down a mathematical expression for it. We don't know what that is, but that doesn't mean that it's not.
It's like hypothesized, like hypothesized utility landscapes, which can explain that, like the symptoms or the things that are seen in the autistic individuals.
I think so, yeah. So that, I mean, again, like with all modeling, all models are wrong. It's only whether it's a useful tool to help us understand the disorder. But
Yeah.
you're totally right. It's limited in that way.
Okay, I have another question. Also, I mentioned like how you said in one of the slides towards the last latest slide, you said the decoder is working better for the knocked out mice individuals' neural responses more than the normal samples. So I was wondering how does this decoder work? And maybe the variability in the population responses is there and is still lower than the knocked out mice, but for some reason this decoder is not capturing this variability in the normal participants.
Yeah, that's a good question too. So yeah, and I didn't explain the decoder. So the decoder, it's a binary task. So in principle, you could use logistic regression or something, but that didn't work very well because we don't have very many trials compared to the number of neurons. With many more neurons than trials, so it was sort of pretty constrained. So the decoder we did use was convert the activity into binary spikes. So a neuron's either spiking or not spiking.
Yeah.
Then estimate the mean firing rate of that neuron, and that's its sort of parameter for its firing rate.
Okay.
And then given an activity pattern of 1s and 0s, you can say what's the probability of that activity pattern, assuming the neurons are independent, just by multiplying the individual probability. So it's a very simple model, but easy to fit. So like I said, maybe we can build a better model with correlations, for example, pairwise maximum entry model or something.
Yeah.
That'd be better.
Cool. Okay, thank you.
Okay.
Great. So next question is by Maheva. I'll just invite on screen. And it's probably similar because it's again asking how is the utility of a circuit defined.
So
I guess we...
yeah, I mean, I totally agree.
I guess everybody is concerned about this. Yeah. Okay, so I'll probably skip this one because it was answered before. So there's a next question. Let me invite Jenny Basu on the screen.
Okay.
So it's on neural variability in response to sensory stimuli. Is it proportional to the severity of the symptoms? Example, hypersensitivity.
I don't think it's ever been measured. We did measure it. So the mice would,
Sorry, I'm here now.
Oh, hi.
Yes.
So I actually so the question is...
If you want, just go ahead and ask. Jenny.
Okay.
So the question is basically, is the greater variability sort of coming from the fact that the individuals with autism might have a different grade of severity of symptoms? So because we define hypersensitivity as not typical sensitivity, but there might be a gradation in there. So is your variability coming from the fact that you have a wider spread of symptom severity? Again, like I said, it's probably a stupid question, but
no, it's a good question. It's kind of getting at the heterogeneity across people, I guess, and it would be nice if the trial-to-trial variability, one of the things we measured, correlated with the symptom severity by some cognitive measure of symptom severity.
Mm-hmm.
We definitely haven't looked at that. The mice in our case were just, or I say our case, the UCLA Labs case, they didn't measure any behavioral anything, and the mice weren't doing a task. It was very passive. They're awake, but they weren't doing anything. And as far as I'm aware, nobody has looked at the same thing in autistic people, which would be a lot easier because usually they will have been measured. But I haven't seen it. But somebody may have done that, and that's a really good idea.
thanks.
Great.
I don't know how to.
Thanks, Jenny. Okay, so we have a question from Andrew Lin, and let's invite him on screen.
I see Elizabeth Mill has replied to the person I said at the start. She's tried to look at this in terms of EEG, variability in cognition, behavior, and they never seem to have a relationship with severity. There you go. Okay. So I don't know if that means that the theory is a bad one or not, but the result's real, and we'd like to get to the bottom of it. It's a difference. Sorry, I interrupted.
No, it's good because it's related to, this question, the earlier one. So I've invited Andrew in the meanwhile. Hopefully we see him. If not, we'll just answer it. There he is.
Hi.
Right, so my question is, you talked about increased trial-to-trial variability in the fMRI mice. Do you think this represents noise, like the sensory responses are somehow unreliable? Or does it represent variability between trials in some kind of top-down regulation? Like the internal state of the animal is changing in some unpredictable or unknowable way, and this is what's causing the variability?
Really good question. Don't know is the short answer, but to assess that, we would we don't know if it's noise or sort of deterministic variability. To assess that, we'd either need more behavioral measurements, as other people have tried to, or other people have shown, like Carson Stringer and Marius Pastorio have shown that a lot of the neuropopulation variability can be explained by facial configuration, which maybe is indicative of some high-level brain states. The one way we can get at that we've talked about but haven't yet done the analysis is to compute the noise correlations of the neurons in the different activity conditions and try and ask how much of the covariance or the variance in the population is due to the stimulus versus the background activity. And maybe that fraction is different between the knockout and the wild type animals. And that's analysis we have planned, but haven't done that. But it's only kind of indirectly answering your question, which is super important. I don't know if it's noise. It's just observed variability.
Great, cool. Thanks, Cian. So next question is by Gabriela. Let me invite her. Let's see. It does take a while for people to come on. But we can read the question in the meanwhile. So can convergence, robustness, dependencies, and multiple solutions also be observed in the cognitive and behavioral level? Yeah.
I don't know. I think, to be honest, I don't know much about the cognitive science literature in general, and I do know that the redundancy thing is a statement about parameters of the brain, and perhaps people have tried this, but I don't know. But what you could do at a cognitive level is build a mathematical model of the agent or the person, their behavior, and usually those models have parameters, and there'll be some dependencies between those parameters. So I'd be shocked if nobody has assessed that ever. I'd be surprised if anybody has looked at that in the case of autistic people, but I'm sure you could do it. And it would be interesting to see if we could relate it to this lower level neural components viewpoint.
Yeah, so maybe just following up on that, I could ask something, which is,
Yeah.
come online also, just to be democratic. Yeah, so I mean, how would this now relate to your current experiments going on? Would you, sorry.
Yes, every time I talk about this, I should tell you this is 2 talks because it's hard to give a solid answer to that.
Right.
What I did during my postdoc was I had a previous collaboration with Carlos where I built a computational model of this bit of the brain, layer 2-3 somatosensory cortex, and I tried to look at sensitivity of changes in the parameters to some aspect of circuit function. So I think that would be a route to doing that. Try and relate the neural activity things we have to the circuit components via a computational model. Or the other way would be if they could measure some of those things in the same brains of the same animals that they're recording the activity in. But that's quite tricky, but it would be doable.
Are you also are the experimentals also looking at, say, some tasks which would give, in some sense, a clearer indication of, say, real-world utility rather than just what we might think of neurology?
Yeah, definitely. So in Carlos's lab, they had a Nature Neuroscience paper last year or the year before where they, in the same mice, they found deficits in a visual discrimination task. So, sorry, visual acuity task where the mice had to distinguish between different shape or different bars. And they found that they could, or that was correlated with PV neuron activity, and they could rescue the behavioral level phenomena by elevating PV neuron activity. And then they even went and shockingly did find the same behavioral deficit in Fragile X patients, in real people. So that was very impressive, and that would be a very nice way to go. So I think more and more there's a search in the autism research community for tasks sensory tasks that map between the humans and the animals. So then that's an easy thing to measure in animals, whereas saying a mouse has autism with social communication deficits and things like that is very hard. So if we can find these sensory phenotypes, it might be much more tractable for addressing your question.
Cool. And then at the same time, these things have to be simultaneously measured, the different parameters and the different neural activities and their performance on tasks. To then
it's feasible, but
yeah.
the big challenge is, like I said in the talk, that the real circuit's very high dimensional. The parameter space is very high dimensional. So measuring lots of parameters jointly in one animal is a big challenge, irrespective of whether you're also measuring the activity. So that's the biggest limitation. But like Eve Marder's shown, that doesn't mean that it's not real or unimportant. It's just hard to measure. So the challenge is going to be making progress according to that viewpoint. But I'd rather like face up to the problem and, you know, think about it rather than pretending it doesn't exist.
No, that's cool. I mean, it's great to see that things are there's a lot more planned. Yeah, and so I'll invite for the last two questions. Oh no, there's one more come up. But anyway, I shouldn't say oh no. But yeah, I mean, so it's by Anna. I'll invite her on screen. Let's see. If not, we go ahead. How did you decide which region of the brain to analyze? Are you going to repeat the experiments in other regions? Yeah, we have.
Hello.
Yes, you said. And why?
That's an easy one for me to answer. It's just the part of the brain that Carlos's group study. So they know a lot about that in development. There's also a practical reason, is that the layer 2-3 cortex is very superficial, very near the surface of the brain, so it's the easiest to do the imaging in. It's much harder just to get a microscope deeper into the brain, but people are trying. But we, well, at least I'm not planning on doing those experiments. And as far as I know, Carlos's lab mainly focused on this with the brain. They also look at other bits of superficial cortex. So that visual acuity discrimination task that I mentioned, they did that study in visual cortex, but they didn't look at variability in that case. But I guess we could try and reanalyze the data. That's a good point. But I don't. One thing I like about the variability idea from Elizabeth Milne and others is that it's fairly agnostic to the bit of the brain you're talking about. Hopefully you could in principle apply this to almost any part of the brain and make some progress. And it kind of just feels like it makes sense compared to some of the other ideas in the field, or excitation inhibition imbalance, which sounds generic too. Hi, Fliss. But that's a circuit. That's a circuit component viewpoint rather than a circuit function viewpoint.
Great. We have Felicity on screen now for the next question.
Hi, Cian. Yeah, I was just interested in the data that you had with the 5 Hertz and 10 Hertz simulation, and you have the firing rates of different cells. And I just noticed that there seemed to be a drop-off, which I guess comes down to synaptic fatigue. But I was wondering if that was something that either you're measuring also in the two models, in the FMR1 and the wild-type model, or if you are calibrating for that, so that that's not something that is skewing your results.
It could, yeah. I think Beatrice has looked at it. And so, well, first of all, Carlos's group had a Journal of Neuroscience paper about 2 or 3 years ago where they directly looked at that adaptation, and they found that the Fragile X mouse had less adaptation to stimuli.
Yeah, I assumed it was a homeostatic thing going on potentially, but
yeah, well, it's a very short time scale, but yeah, you're right. Yeah, so it seems like they have less adaptation to repeated sensory stimuli. And in this case, that decoding analysis, we just ignored it, but you could build in a model where you factor that. That would be true. To be honest, I haven't thought about how that would affect our results. Maybe Beatrice has. Sorry.
Okay, thank you.
Yeah, thanks. And we go for the last question by Nada again. It's on the experimental strategy. Let me see if I can. Thanks, Felicity. And yeah, let me invite her again.
should I read it, or will I wait for her?
I mean, you can go ahead and so yeah. It's
Okay.
regarding experimental.
So-
Yeah, sure.
Other question though, sorry, yeah. So the last question I was asking about the frequencies that you picked for flicking the mice whiskers. Do you think, or have you met, like, do you think it's worth trying other stimuli frequencies? And I know that the experimental recordings and doing the whole experimental setup is like, it's not the easiest thing to do, but have you considered investigating other frequencies and how this would affect the variability you are looking for, or the variability within the neural responses for knocked out mice versus the natural mice, or not?
Yeah, it would be nice to have more stimulus categories, or maybe a continuous stimulus, definitely. I'm not sure what... I'm sure it would... Well, there's probably some questions we could address with more complicated stimuli, but at least for the things we cared about in this study, the binary stimulus was enough. Because like you kind of said, the big challenge is getting enough trials to measure, but it can be done. I mean, other groups.
I was asking, like, what's the motive or the incentive for choosing like 5 hertz, 10 hertz, or is it just random? Like,
kind of random, yeah. I think other things you could vary are like how long the stimulus was, or the amplitude of the stimulus, so how big the whiskers are. They're natural things that presumably happen for natural whisking, but we chose these 2 frequencies because the natural whisking frequency, if the mouse whisks its own whiskers, it's usually on this 8 hertz-ish range. So we think this should be fairly physiologically relevant. It's a fairly abnormal stimulus because we're just grabbing all the whiskers and wiggling them together, and the mouse is passive. But hopefully the types of stimuli the brain's getting are fairly physiological.
Okay, makes sense.
Yeah, but this is a crucial question, because it could be totally different in a different regime.
Yeah. Okay, cool. Thank you.
Thanks.
Thanks, Nada. And yeah, let's thank Cian again for the wonderful talk and all that.
Thank you very much, everybody.
For the questions. Yeah, we've already seen some appreciation in the chat box, and and I guess so. Thanks again, and
thanks very much,
you.
Eleni,
Yeah, sure. Yeah, thanks, Eleni, and
Cian.
Cian.
Bye.
Ciao then. Bye.